welcome, bienvenue, Annie. I am very excited for what we are about to do tonight. To introduce myself, my name is Amy Henson. I'm a senior scientist at Science North in Sudbury, Ontario, Canada, and we have got an amazing evening lined up for you tonight. We're finishing off Earth Week this week and the celebration of looking at our environment and how all the things that we can do and how we can help our planet every day. And there's nothing more than I can think about than helping our planet than science. And of course, why wouldn't we be talking about science? So we have a great evening tonight where we have partnered with the Vale Living with Lake Center to really show you some amazing research and amazing science. But where we are located here in Sudbury is a very, very important place. And we really always want to acknowledge exactly where we're located and our location. And Science North and Laurentian University are located on the traditional ancestral lands of the Atikameksheng and Anishinaabe. Sudbury is also on the traditional lands of the Wanapitei First Nation. And we also want to acknowledge the Métis Nation for their cultural and economic contributions to our area. These lands that we sit on are in the Robinson-Huron Treaty area of 1850, and it's a great reminder to always remember that we, every single day, are all treaty people. It's important for us to be thinking about what treaty area that we live in and that we actively take part in every single day. So let's take a moment to appreciate the land that we're on by not only acknowledging the traditional territory where Sudbury is, but also your own traditional territory and treaty area that you're located in. So to start tonight, I want to introduce and bring on as my guest host this evening, Dr. Chantal Barriault. Hi, Chantal. How are you?
Hey, I'm really good, Amy. Nice to see you, and I'm super excited about our event tonight.
It's going to be amazing. Now, Chantal is no stranger to Science North whatsoever. In fact, we call her a blue coat emeritus. She was a blue coat at Science North for many years before she started a program at Laurentian University called the Science Communication Graduate Program. And that program is based out of the Living with Lake Center, which is just down the road on the shores of Ramsey Lake here in Sudbury, just like Science North. It's not too far. I can walk or bike there. It's just a hop, skip, and a jump. And so, Chantal, maybe tell us a little bit about the Vale Living with Lake Center. And this thing, it's kind of a mystery. People may not have heard of this before, but the Cooperative Freshwater Ecology Unit. So tell us a little bit about that.
Yeah, that's right, Amy. So just across the road from the buildings of Laurentian University, you may have driven down there and seen the beautiful lake center, the Vale Living with Lake Center. And that's where the science communication program that I run with Science North, actually, and Laurentian, that's where many, many scientists and researchers and students hang out and do lots of awesome research and discover all kinds of things about our environment, about the fresh water that we live around, the fish that live in it, perhaps the contaminants that are in there. And there's the group of people call themselves the Cooperative Freshwater Ecology Unit, or co-op for short, because it's a pretty long term, isn't it? So we call it the co-op, and there's people in there from government, from university, from industry, and they all make up this cooperative unit, and they work together to better understand and find solutions for our lakes, our rivers, our forests, our soils, our fish, and all the things that make living in Northern Ontario absolutely fantastic. So yeah, the co-op unit down the road.
Down the road. And I tell you, it's like a second home to me. I love going to that building. It's where I did my master's research. So it's definitely a very, very special place. The co-op unit has always held a very special meaning within here in Sudbury. And so we're excited to celebrate this because yesterday on Earth Day, the Cooperative Freshwater Ecology Unit held their annual general meeting. And really, meetings can often be boring. But not when it comes to this group. They spend their day just talking and celebrating all the amazing research that goes on there, discovering what everybody's been up to over the past year and how far they've gotten in their research. And one of the biggest highlights for me is hearing from both students and listening to their up and coming brand new research, but also hearing from many of the veterans that do their work in the center and have done that work for many, many years, an important ground-laying work that is in the environment and freshwater ecology here in Sudbury. And we all know that Sudbury has led the way in environmental remediation, and I mean, what a better thing that we could do tonight to celebrate Earth Week. So Chantal, why don't you tell us a little bit? We're kind of going to go through this tonight. We've taken excerpts and snippets of this meeting that they've had and really gotten the best stuff out there for you to see tonight. Chantal's going to lead us off. And she's going to take us kind of close to home, because close to home, I think, is a great place where to start. And you've never, and I'm sure that you've heard all about, if you live in Sudbury, you're very familiar with Junction Creek. And Chantal's going to share some great research coming out of Junction Creek this year.
That's right. So one of our up-and-coming researchers at the Lake Center is Mackenzie Russell. She's a master's student with us, and she's investigating what is actually happening in Junction Creek. Specifically, she's really trying to understand what happens if you have periods of drought, because with climate change, this is happening more often. We're getting extreme events, right? From flooding to drought, all things that we don't necessarily experience more than every 100 years, but now because of climate change, it's happening much more regularly. So Mackenzie's research is about what happens in periods of drought to our city's creeks and riverbeds when they do dry up, and why would that matter anyways? Well, before we listen to a snippet of Mackenzie's research presentation, I'll tell you that when water levels drop, it actually can kickstart the processes that drop pH. So it makes the environment or the soil or the riverbed or the creek bed more acidic when that happens. And of course, more acidic environments are really hard on creatures, right? On the creatures that live there. And it also, by drying it up like that, it also makes metals in that environment become more toxic, because they're already there, but they come to the surface and they can become more mobile. So we need to learn more about drought and the processes and what happens. So I'm going to let Mackenzie Russell tell us a little bit about that research.
So this is a really important process to understand, both as climate change continues to increase the frequency and severity of both drought and flood events, and also regionally in areas that have received high levels of sulfur and metal deposition, such as Sudbury. So our objectives were, number one, to help inform drought management practices as well as land reclamation practices. With Junction Creek running right through the city of Sudbury, right through downtown, obviously controlling stream flow during these drought and floods is really important. One of the proposals or ways that this is often dealt with is removing or dredging out sediments. So we want to make sure that this is dealt with and that these sediments, if needed to be removed, don't lead to these reacidification and metal release events. In terms of land reclamation practices, we're interested in the use of watershed liming, as John mentioned, because it has been used in the region to improve water quality and reduce acidity. So whether that application of liming before a drought or drought season could improve this drought response. So my site specifically is the Nickeldale Dam and Reservoir area located in the Frood tributary. The Frood tributary on a whole, but this site in particular, has received high levels of prolonged historic contamination of both metals and sulfurs from mine effluent. We chose to focus on more wetland ecosystems because of their importance for watersheds on a whole in nutrient cycling and storage, including that of metals. Our analysis included looking at the total and dissolved metal concentrations of the water post-drought, as well as the dissolved organic carbon concentration, because I think it was, as Canali mentioned this morning, we know that organic carbon is really important in both the mobility and toxicity of many metals. And those chironomid midges, we were primarily looking at percent survival in each treatment, and also hopefully looking at total metal concentrations of their tissues. So I'm currently kind of in the early results interpretation phases, still receiving more and more data, but early indicators do show that liming does reduce the level of reacidification happening post-drought and potentially decrease the concentrations of at least a few of the metals we were looking at. So obviously we're interested in helping mitigate the negative impacts that we might see from these sediments during a drought, and also just adding to knowledge about regional climate change impacts that we can expect and hopefully better adapt to.
Excellent.
That was great, wasn't it? So yeah, exactly.
Amazing research, Mackenzie. That to me, that's so interesting, because I never would have combined those two things together, where you have a pH drop, right? And now you've got metals that are coming to the surface. Like those two things maybe don't naturally correspond in my brain, but that is really neat, and how applicable to other areas, not just Sudbury, right? So even though that research is being done here, it's applicable to other areas across the world, really.
Absolutely. And the fact that we can use liming, which Sudburyans are familiar with, if they've grown up here, we lime to reduce the acidity of soil. And if liming is actually going to work in, liming riverbeds and creek beds so that, that toxicity doesn't get out of hand, I think it's just really important research to do. So
Fantastic.
Way to go, Mackenzie.
All right. Well, let's move on, because this kind of connects to our next researcher who we have. And this is Jade Dawson. Jade is a master's student at the Lake Center. Now, Jade is actually looking into the long-term effects of reducing the sulfur dioxide emissions on our lakes and applying that to the land reclamation processes that we've had here in Sudbury. So again, those of you who lived in Sudbury, remember, this is what we had, right? Is very, very, very acidic soil, and applying lime to that soil and the black rocks that surrounded Sudbury, and that lime will help neutralize that acidic soil. Then planting grass seed and eventually trees and regreening our landscape. And this whole process, I think, is really amazing. It has its own name, and it's called the Sudbury method. And it was a methodology, right, developed right here in Sudbury. And so Jade's going to talk a little bit about, the Sudbury method and how his research and how their research relates to that. So Jade's investigating how the Sudbury method impacts the recovery of our lakes, and that relationship. So how does life reestablish itself in the recovering Sudbury lakes? What are the best tools to detect these signs in these lakes, and how do we look at how those lakes are recovering from that acid damage and the mining practices that we have here? So let's take a look at Jade's research.
So to do this, my study examines recovery at two scales. We have the whole lake scale that's more commonly used in rapid biomonitoring sampling, and the delta scale, which is the confluence zone between the river and a lake. So as you can see from this image, the river creates what's called a sediment delta from the soil and organic materials that it transports to the lake. And this provides a biological hotspot and subsequently a dispersal site for aquatic life, given the enriched nutrients and primary productivity at these delta zones. So in my study, I've included an intermediately damaged lake that did not suffer vegetation loss or acidification. This is Nelson Lake out in the Valley. I also have a treated lake that was previously severely damaged, Daisy Lake. And finally, I have a severely damaged and untreated lake, Baby Lake, that is recovering due to the clean air effect and natural revegetation only. So using my 3 study sites effectively, I'll be working across a gradient of both catchment impact and treatment. So this will allow me to understand the role of delta sites in influencing lake recovery and how this might change over time. So to assess the chemical recovery, I took water samples at the delta sites and as well from the deepest part of each lake at historical sample sites. And these were analyzed for a suite of nutrients, pH, and heavy metals. And for the biological recovery assessment, I conducted surveys of several different aquatic organisms, all the way from microbes up to fish. So one of the main water chemistry parameters associated with the smelter impact is the concentration of nickel. So the graph on the left here represents the nickel values determined from the lake water samples. And when you compare this with the graph on the right of the nickel concentrations from each of the delta streams, it becomes clear that the whole lake sampling approach does not effectively capture the variability of within lake nickel levels and tends to underestimate the nickel concentrations at these key dispersal sites. The main findings of our study so far highlight how critical small-scale sampling efforts like those of deltas are in detecting and assessing early-stage lake recovery. So these results emphasize the important role that delta sites play in whole lake recovery, and future biomonitoring efforts and site selection should target deltas specifically to accurately model lake recovery, at least until the within lake variation of these delta sites becomes negligible.
So great research again by one of our own, by Jade, who's really focusing on that delta. I'm hoping our viewers really saw what a delta is. It's literally where the river where a river or a creek pours out into into a lake, and it creates all of these sediments. And their research is suggesting that we should really be paying attention to that, because of course that's coming off, land and from rivers, and is probably bringing things like in the sediment, right, like metals or what have you. So by studying the deltas, you're getting a better idea of whether or not that lake is recovering. So really, really important research.
Yeah, it's kind of one of those things where I always think of, an eyedropper, right, in a glass of water. So you have this big glass of water, and and then all of a sudden you add like a little bit of drops to it, right? And just for those drops enter, the concentration of whatever you're putting in there is going to be a lot higher in that area than it is going to be in the rest of your glass of water. And it's kind of working the same way in a lake system that way, where you've got this delta coming in. It's bringing all kinds of stuff with it, and we need to think about how we sample lakes in different ways. And that's some really, really promising research, Jane.
Yeah, exactly. Well done. Well, we're all if we grow up in Sudbury or Northern Ontario, we grew up with lakes, so we're very familiar with lakes. We're also probably very familiar with fishing. Who doesn't love to go fishing? And I'm looking outside now, and I'm missing out on a really nice evening, but I'm happy to be here with everybody. So the next group of researchers that we're actually going to share with you what they're up to is the Lesgord Lab, and Dr. Gretchen Lesgord is the lead researcher in this group, and she's going to be presenting next for a few minutes with a sample from her students Adam Lapage and Calvin Kluk. So what this group is looking at is they're trying to study how and why metals accumulate in wild fish across the province, because they're trying to inform future environmental monitoring programs. Like when we pay attention and we monitor, like what are the levels of metals and are they okay to eat, right? What are these fish okay to eat? Because bioaccumulation means as the fish continue to eat within a lake that might have metal contaminants in it, that metal can accumulate in their skin tissue or in their muscle tissue. So that's what Dr. Lesgord's lab is looking at. So you'll hear, one of the things I wanted to warn our viewers about is, not warn, but just give you a heads up on some terminology, is that Dr. Lesgord and her students often say species. And I always think of species as animals. And what they're talking about here in this case is actually species of metals. So they're looking at arsenic, mercury, and chromium. And when they say species of mercury or species of arsenic, that's what they mean. They mean different forms of mercury or different forms of arsenic. And depending on which form the mercury or the arsenic take, that will make it more or less toxic to human consumption. Or to animal consumption. So it's really important for them to differentiate the different types or species, you'll hear, of arsenic and mercury. And the last word I'm gonna just give you a heads up about too is subsistence fishing. So subsistence fishing is not sport fishing. It's fishing basically to feed yourself and your family. And if you weren't fishing, you would not have a food source. So there's a lot of communities in the far north of Ontario that do subsistence fishing, and it is one of their primary sources of food, right? So for them to consume a lot of fish that may have high concentrations of metal, not a good idea, right? It can become quite toxic. So with that, let's tune in and listen to what Dr. Lesgord and her students are up to.
We know that mercury, arsenic, and chromium are the three elements of concern in fish in Ontario. And we know that because the province does a pretty thorough job of monitoring contaminants in freshwater fish. They analyze contaminants in various fish from over 2,000 water bodies and report on those concentrations online and in a book. But all of that work, the monitoring in our past work, has been done on total concentrations of these elements. So they can all exist in different species or in different forms. So different species of these elements differ in their chemistry and therefore in their behavior and their toxicity. So for mercury, we generally talk about two species that are important in environmental sampling: inorganic mercury and methyl mercury, with methyl mercury being the toxic form. Arsenic's a little more complicated. There's a number of different species that can occur, including several organic-based arsenics, such as arsenobetatine, which is less toxic, and then inorganic arsenic forms, arsenate and arsenite, which are carcinogenic and a concern. For chromium, we're back to 2 key species in the environment that we measure, trivalent chromium and hexavalent chromium, which hexavalent chromium is a well-known carcinogen as well. So when we do speciation analyses, what we're doing is separating out these different forms of these elements and quantifying them individually. And then we often compare that to a total analysis, which lumps all the species together in one value. So both of these analyses have a place when we're trying to understand contaminant dynamics. They're kind of 2 sides of the same coin. And you're going to see that in the next 2 presentations from our 2 great students who are working on arsenic. So Calvin Kluk is using total arsenic and looking all across the province at what drives arsenic accumulation in fish.
The Ministry of the Environment, Conservation and Parks and other researchers have done a great job of monitoring contaminants in fish across the province. Reasons why arsenic concentrations are higher in some water bodies is not fully understood. A couple of the main goals of this work is first to combine several valuable data sets. So really what we've been spending a lot of time on to this point is gathering and repurposing data, which has already been collected, and aggregating it into a more usable format, which could be used by others and added upon in the future. And secondly, we're hoping to provide information for future monitoring efforts. So because analyzing fish for arsenic can be quite costly, identifying which physical and ecological variables are related to these higher concentrations of arsenic in fish can help to inform which water bodies to focus monitoring efforts on. And this ends up benefiting anglers and subsistence fishers across the province.
So that's a very broad study design. And then Adam Lapage, who was a BSc student, now starting his master's on arsenic speciation in fish, has sort of a more targeted or refined study question.
While we often may think of arsenic as a singular thing, it actually occurs in more than 100 different forms naturally in the environment, referred to as its species. However, contaminant monitoring and research programs will still most often measure all of these species together as a total arsenic value due to the ease of this standardized analysis. But this has its limitations, most notably that it does not account for the wide range in toxicity that exists among the different arsenic species. So, one solution to the limitations of total arsenic analysis is arsenic speciation analysis, which allows us to directly measure the concentrations of the different arsenic species. So that leads us to the main goal of my undergraduate thesis work, which was to test and validate a method for its performance. And so I'll be looking to improve performance of this method for inorganic arsenic, and then apply the method to more fish from more species and also from more lakes, so that we can gain a better understanding of arsenic bioaccumulation patterns in freshwater fish, especially those that live in contaminated sites. This method will enable us to further study arsenic speciation patterns in the Sudbury area and beyond.
Really what the research is trying to help do is answer this very complicated question of, are our fish safe to eat? And that's not a question that we alone will come anywhere close to answering, but we are trying to help with aspects of that question. So what I would say are our past and future goals with contaminant work, the way I see them going is first, we're trying to identify contaminants of concern in wild fish with a particular emphasis on subsistence fish. Second, we're trying to explore variability in those contaminant concentrations and drivers behind that variability. We're trying to refine monitoring and consumption guidelines by applying speciation analysis where possible. But in some cases like chromium, it's quite challenging. We acknowledge that fish is a very healthy protein source and is an important aspect of people's diets. And I'd like to see us incorporate more of those health benefits into our work as well, including some of the fatty acid work. And overall, the goal, as I see it, is to try and help inform future monitoring and consumers about potential risks, to enable them to make more informed decisions about what they're eating and what they're feeding their families.
Excellent. I mean, that is something that, as a fisher myself, right, that we use in my family, is to whatever lake that we're going in, we use the guide that the Ontario Ministry of Natural Resources and Forestry puts out, so that we know whether the fish are safe to eat, right? So that is something that, I do as a fisher every time I go to a new lake, and those values change from year to year quite often.
Exactly.
Yeah. Really, really important work, and especially when we're relating it to subsistence fishing. So great.
That's right, because if you eat, like, you know, Amy, maybe you're going out and having, you know, maybe a fish or two a week in the summertime or something like that. But other communities, maybe if you're catching 20, good for you. But others are literally having, like, you know, we have to maybe put a limitation, like you can only have 12 fish a week of this type because the levels of arsenic or mercury are too high or something like that. So that's really important work that Dr. Lescour's lab is doing. So
absolutely.
Yeah.
And I mean, I love talking about fish. Fish are my favorite animal, just so everybody knows. They're my favorite animal. But we're gonna take a little bit of a look about with some other stuff. And I brought something with me, Chantal. Are you ready?
Yeah, I'm ready.
Are you ready? Because
You're the prop person.
I brought props. This is really important. So water, nice and clear, right? Take a drink. And then I made myself a lovely cup of tea.
Nice brown.
But it's brown, right? Water, brown. What does this have to do with science? Well, let me tell you, because we have a wonderful scientist who's working at the Living with Lakes, Kanali Gahil, who is doing their master's research. And she's investigating the browning. Oh, yes, the browning of lakes in the Sudbury area. And it's a really interesting thing. So when we take a look at lakes, and if you've traveled around the Sudbury area, you've probably seen two different types of lakes. Gone to see a lake that you look at it and it's, you know, beautifully, brilliantly blue. The water is super clear. You can look straight to the bottom. And I mean, visually, it's stunning. Like
some of the lakes in Killarney, right?
Some of the lakes in Killarney. Like, and I mean, how many of us have seen, you know, the Instagram photos, and like, they're beautiful. Like when you look at them, they're gorgeous. But one of the things about them is they are biologically dead. There's not only is there no fish in there, there's really no life at all. There's no insects, there's no zooplankton, there's no phytoplankton. There's nothing in there because nothing can survive the acidity of those lakes that are there. And it's the color. So when we get color in lakes like this tea brown, that's when we start to realize that I bet you there's something in there, and I bet you this lake is a little bit more alive. So what's happening now that we have, of course, come a long way in our reclaiming of our landscape is that our lakes that were once clear and biologically dead are going through a process called brownification. They're bouncing back and they're coming back. And Kunali is going to explain to us a little bit about brownification, but also her research that's associated with it.
So lake brownification is a change in the visual color of water due to an increase in organic matter runoff into the lake. This organic matter contains color dissolved organic carbon, which is often referred to as nature's tea, giving our lakes that tea-colored appearance. It is an important sign of recovery of clear acidic lakes. So this brownification causing DOC is a master influencer. It has physical, chemical, and biological effects on lakes. Physically, it can cause heat attenuation in the surface waters, causing changes in the thermal habitat structure of our lakes. It also acts as a sunscreen for various biota and prevents photo bleaching of fish, which used to be a big problem in many clear acidic lakes in Sudbury in the past. Chemically, DOC molecules consist of fluic and humic acid molecules, and these are large negatively charged molecules that often attach to toxic metals and lock them away. And biologically, it can boost productivity in lakes, bringing in terrestrial nutrients. But if DOC surpasses an optimal level where there is not enough light for photosynthesis, then we have reduced productivity. So my study area consists of 82 lakes in the Sudbury region, and the lakes are divided into two main groups, acidified lakes and reference lakes. We added a second set of reference lakes known as the Turkey Lakes, and this set of reference lakes would be beneficial as they are located outside the Sudbury region. Now, all of this data was collected by the Ministry of Environment, Conservation, and Parks. So my project consists of studying zooplankton community responses of these 82 lakes and the Turkey Lakes and mapping their responses to various environmental variables, including DOC. And I hope to infer the health of the ecosystem by explaining bottom-up and potentially top-down food web changes and management of a healthier lake ecosystem. So one objective was to try and understand if the current zooplankton community characteristics are shaped more by the acidification histories of these lakes or due to other contemporary differences among the lakes. Overall, DOC is an important influencer for the Sudbury region lakes, and monitoring our lakes helps us understand contemporary biological relationships and helps us keep track of emerging concerns that may be magnified by brownification.
There you have it, brownification of lakes.
I love it. I love it. And you know, one of the things about all of this is when we do this type of research, it just doesn't exist on its own, right? And one of the things Chantal is really looking at is all these different factors that factor into our environment. So whether it's an anthropogenic factor, whether it's an urbanization as an example, or chemical or physical factors in the environment. And when we do this type of research, it's so important to take all of these things into account. And that's what really, I think, really builds this robust sort of understanding of how we're coming back. Is we're not just looking at one little thing. We're trying to bring in all of these different ideas together to really get a holistic picture of what's going on around us.
Yeah, exactly. And the next researcher that you're going to talk about, Amy, is also talking about lake recovery, but in particular, how our lake recovery has been helping lake trout. Absolutely. So I'm going to hand it over to you for that one.
Yeah, and I mean, again, I love fish. I can't help it. I'm a marine biologist. I mean, I really do. So Jasmine Luce-Fillon is a researcher as well, and she is looking at what we're going to call trout repatriation. I love this word. It is such a great word. It's, you know, I'm like, it's got a lot of oomph to it, but I love it. And you know, trout and lake trout especially, I mean, it's definitely one of my favorite fish to catch and get on my line. And, you know, you go seek out the deep lake where, you know, you think you might find them along a rock wall or something like that. But trout is a really important sport fish here in Ontario. And of course, with the acidification of Sudbury lakes, those trout basically pretty much were, you know, extirpated from our area. They
vanished.
They just don't do well in that type of environment, period, let alone like with stressors, let alone a lake that could be dead, as we sort of described before. And so we've been, you know, working to bring many different species back to our lake systems, including lake trout. And this repatriation process Jasmine is going to talk about, I think you'll find it really interesting. If we can get the video.
You may be asking yourself, what is repatriation? Well, the repatriation of wildlife is to improve the status of its population by reestablishing previously extirpated or damaged populations to their native habitat. The objective here is to end up with self-sustaining and reproducing native populations in areas where they were previously lost. This is a problem we know a lot about in Sudbury, where over a century of mining and smelting led to the acidification of an estimated 7,300 lakes in its acid deposition zone. Of all the sport fish in the area, lake trout were the most heavily impacted, nearly extirpating resident populations from the area. But as you've heard today, emissions have since reduced over 95%, and lake trout populations are starting to recolonize. Which leads me to my own research project objective. Lakes in Sudbury are on an ongoing recovery trajectory from this damage, and my objective is to determine what key factors play a role in the success or failure of these rehabilitating lake trout populations. To evaluate this, 31 lake trout lakes of varying degrees of recovery were surveyed over the last 2 summers. A full broad-scale fisheries survey was completed on each lake. Plankton communities were sampled, dissolved oxygen and temperature profiles were taken, and water samples were collected. I'm currently using 2 measures of population recovery in my analysis. The first is lake trout biomass catch per net as a measure of population recovery status. And the second is evidence of natural reproduction. So the first potential relationship I found is between lakes with naturally reproducing lake trout populations and total fish species richness. So what's interesting here is that lakes without recruitment had slightly higher species richness values in comparison to lakes with natural reproduction that had generally lower species richness values. But this is in line with the literature that lake trout do better in more simple food webs. Next up is the relationship between the presence of yellow perch and lake trout catch per net. So the expected result here was an increase in lake trout biomass with the presence of yellow perch, since perch are a common prey for lake trout. However, the trend seems to be going the other way with less lake trout biomass in lakes with perch. This could be due to lake trout primarily feeding on zooplankton and not fish, something we found when checking their stomach contents in the field. And finally, I want to touch on the potential importance of lake size in lake trout recovery. This figure looks at the relationship between naturally producing lake trout populations and maximum depth of the lake. Here we're seeing a slight increase in lake depth with naturally reproducing populations, likely because a deeper lake means more habitat for the cold and deep water lake trout and less overall competition. So in conclusion, this idea of fish repatriation is a fairly novel one, and I believe Sudbury's recovering system makes a great model for the better understanding of what factors play a role in lake trout recolonizing these previously damaged areas.
And this one,
you might be able to eat lake trout from Sudbury lakes again soon.
You might, and you know, and we sort of think, when we think of, we bring species back, you know, we work to, you know, make the, you know, have species that are mating and reproducing in an area. But the difference here with Sudbury is really that this repatriation of trout is going along with the whole remediation of our environment at the same time. And those two things coming together at the same time is, I think, what makes this particular research incredibly interesting on my part.
Absolutely. Yeah. I loved it. I loved the lake trout, and I'm looking forward to hearing more about what she discovers in her master's thesis. That's right. So we're actually going to switch gears a little bit. I know you love fish, but we are going to switch gears. We're going to talk about bacteria and mining.
Okay, whoa, wait a minute. How can we go from fish to bacteria? I'm like, no, okay. Again, I'm a marine biologist. Okay, well, no, I do know where you're going, because here's the thing. You know, I can talk about fish all day, but when I first heard about this research, and I remember hearing about wait a minute, bacteria and mining together.
Yeah.
I'll tell you, two things that I'm really not that interested in. I want to put it bluntly. Bacteria mining, it's all good. But when I first heard about this research, I'll tell you, oh my gosh, mind blown. So why don't you tell us about how bacteria and mining go together?
Okay. So some of the people in Sudbury and surrounding area might have heard about Dr. Nadia Mikachuk's research already. So I'm going to just briefly tell you what that's about in the simplest way possible. I even brought my own prop this time. So here's my prop. So Nadia's research is about bacteria that, or microbes like bacteria that eat metals. They actually consume runoff from toxic mine waste, like tailings. So I've got my little drawing here. This is metal. This is iron. I don't know if you can see that. I got too much lighting.
Yeah, you got it.
Anyways, I got some little bacteria going around here. And basically the idea is she and her colleagues and her research students and everybody that works in her lab have discovered that there's these microbes that actually consume toxic waste. And they consume the metals. And not only do they consume them, but they can also sort of recover some of the metals that are in there, which is just mind-boggling, right? And they're called extremophiles, these bacteria, because they live in these like really extreme environments of like really high acidity or really, really cold temperatures or high temperatures. But using biological organisms like this to do work for us, which is what they're doing, these little creatures, they're doing work for us. We call that biotechnology, right? So essentially what Dr. Mikachuk's work is creating microbial miners. They're literally mining, right? It's biotechnology by bacterial miners. And they've done this very, very successfully in her lab. But in the lab, you can imagine it's a lab bench, small scale, like a liter or two, think about your milk containers, about a liter. But her vision is to go much bigger than that, right? To have industrial applications, and having, bacteria in a liter isn't going to cut it. So I'm actually going to let Nadia now, I'm going to let her video play so that you can hear about what her vision is for scaling this up on a massive scale. Okay.
And we recognize that there's a gap, much like there was in taking restoration and land reclamation tools out of the lab and onto the landscape. We really need to find a way to take biotechnologies that can help us extract value from mine waste, or reduce liabilities and clean up those long-term legacy issues that stem from a very large volume of mine waste that usually gets left out. We set out envisioning what could be a center for mine waste biotechnology, and what would the need be to fill that gap in connecting different stakeholders between academia, the private sector, and sort of filling that niche of being able to provide companies and technologies to those end users, to the mining industry. We couldn't do this alone, of course. Scientists are not trained in developing business plans or organizational models. We enlisted Stiletto Consulting, who are a boutique consulting firm, but have already worked on a number of industrial and tech parks. And the CEO is was in fact the president of the Canadian Association of Technology and Research Parks for 10 years. So they are very well positioned to understand what you need to do when you want to build a research park like this, that is not just private sector, is not just academia, but one that brings multiple stakeholder groups together. But we were able to get funding in place, and we had started the feasibility study with Stiletto in November of 2020. So I can share with you where we got to and what the consultants worked with our advisory team that included a number of different stakeholders, both within Laurentian and the Sudbury community. So looking at what exists currently, not in Sudbury, but in Canada, there are a number of peer parks, as we could call them, that are already working in biotechnology space, looking at the environmental space, or moving towards zero waste mining and developing those areas of expertise and technologies. So we also have to look at what offerings are there, how we can build collaborations, how, again, we can fill gaps that in specific areas that they may not be looking at. And trying to build that Canada-wide ecosystem and help move those larger, broader pan-Canadian strategies forward. There are a few places that can be a one-stop shop that connects academic research all the way through to commercialization. Again, that is very challenging to do, but if we can have it as a focus, then it is something unique and a value add that this center could bring. Of course, we are uniquely positioned here in Sudbury and Northeastern Ontario in what we can offer in terms of that mining ecosystem. And again, that can help sort of bridge the delays that often are seen in moving these types of technologies forward and building on that Northern Ontario cluster of excellence. There are a lot of technologies out there, but being able to match the right solution to the problem is not always obvious. And so we have to, again, build that network that can help troubleshoot and match opportunities to solutions effectively. And being able to do that well can save time and money and increase the output. Biotechnology is still a niche for them that is not fully accessible. And so genomics tools and biotechnology still need a lot of work to be able to bridge that gap in translating those technologies into everyday best practices or usable tools within the mining industry. Being able to de-risk and demonstrate these types of technologies, not just at the bench, but to be able to pilot them and demonstrate them, can really help provide that opportunity to the mining sector and to the mine service sector that don't necessarily have those facilities or the expertise to do that. Looking, of course, to access the knowledge and talent pipeline is critical, and that's something that I've worked very hard in my team and as part of the Lake Center team to develop excellent HQP that go on to do incredible things. And of course, getting the model right. This is a new area for me, and understanding how do we engage stakeholders, how do we monetize this through commercialization and for the end users or investors. It's really the types of aspects of research that academics are not trained in, and we have to think about those if we're going to successfully see these solutions get to market. Just like John's dream of building the Lake Center and the co-op's dream to build the Lake Center, this is the dream that I have to build the Living Without Waste Center and the Center for Mine Waste Biotechnology.
Center for Biotechnology, Living Without Waste. Love it, love it.
I love that.
Nadia's dreaming big, and one of her current postdoctoral fellows is Dr. Emmanuel Noma, and he's investigating how these little microbiology or microbial miners can actually do that return of value, right? And so he's working on taking this from the lab bench all the way up to industrial, scaling it up to industrial size. So here we are, with this vision of Dr. Mikachuk's to really dedicate a whole center to biotechnology to help us really solve mine waste problems right here in Sudbury.
Right here. Oh man, that I don't know. It's the one thing that I always said I am amazed by. Every time I listen to Nadia speak, I'm always amazed at the work that she is doing, and her moving forward with some really amazing cutting edge things found nowhere else in the world. So amazing. So we have another, we're gonna take a step away from biotech for a little bit, and we're gonna kind of go a little bit further north of Sudbury. So we're gonna move away from our local aspect, because there's a lot of research that happens at the Living with Lake Center that has to do with more far northern Ontario as well, and some really great research that happens with that. So we're gonna hear from Adam Kirkwood, and Adam Kirkwood is studying a permafrost thaw in the far north. And so if you don't know what permafrost is ground, so soil and ground that is permanently frozen for a minimum of 2 years, all year round. It never thaws. It stays frozen, and it's a really important part of the northern landscape of our northern hemisphere here on our planet. And we know that with climate change, that permafrost is melting. So you can imagine ground that's been frozen for 100,000 years is now melting. And what are the issues with that happening? And so Adam's going to talk with us about the idea of heavy metals. Again, we're going to talk about metals. They're an important part of what we look at with contaminants, but the implications of mercury and storage and cycling within permafrost.
Permafrost stores significant amounts of the elements of carbon and also mercury. But as it starts to thaw, this carbon and mercury can become a more active part of biogeochemical cycle. So the Hudson Lowlands is a very unique environment, as we've heard already, and it's of global importance. So it's a very, very big peatland. It has an area of 372,000 kilometers squared. So that's about bigger than the size of the United Kingdom. And there's a very large storage of carbon and mercury in the region. So recent modeling shows that there's clear changes in the expected extent of permafrost. So this is a permafrost map of Canada here, and you can see in Hudson Bay Lowlands we have continuous permafrost. But this new modeled map that came out in 2019 shows that permafrost in the northern Ontario portion of the Hudson Bay Lowlands isn't expected to be sustainable anymore, and there's likely quite extensive degradation. So that's one of the questions that I'm trying to answer in my PhD, is how are permafrost features changing and how quickly? So the first goal of my PhD is to develop a map of total mercury in the region. So again, this being the outline of the Hudson Bay Lowlands, is trying to figure out the actual storage of mercury here. And this is done in partnership with the MNRF, with Laurentian University, Western University, who I forgot to put up here, and WCS Canada. The second goal is to determine how permafrost thaw is going to influence this mercury. And I'm specifically looking at mercury methylation, where microbial communities are going to take inorganic mercury and transform it into organic mercury that's available to wildlife and eventually to humans. So I want to look at how different environments can relate to mercury methylation by taking cores of it, determining mercury and methylmercury concentrations, and then extracting DNA from these cores and looking at the relationship between methylmercury concentrations and the abundance and structure of microbial communities. And then the final goal of this project, continuing on the theme of partnerships, is continuing relationship building with communities and research partner. So in the last few years, I visited Peawanuck quite a few times, and I've been so lucky to work together and how we can continue to do so in the future, and how sharing traditional knowledge in Western science is very important. And of course, building capacity with our research partners. So for me, it's PermafrostNet, University of Laval, and of course, WCS Canada, who I'm incredibly grateful for.
There we have it, permafrost and the impacts of the melting of that permafrost and the potential release of mercury and methylmercury because of the bacteria that and the microbial communities that are basically waking up and starting to have an effect, right? So really important work being done in the Hudson's Bay lowlands, and we tend to forget that Ontario is an Arctic province. Like, we have permafrost in our province, and of course our population is concentrated in the south, but there's communities up there. There's all this land base that is really being affected by climate change, and Adam's up there trying to understand it.
Absolutely, for the betterment of all of us in Ontario, regardless of where we live.
Exactly, and that actually leads me to Nicole Corbière, because she's a master's student. So Adam is a PhD student looking at permafrost in the far north. But Nicole Corbière is a master's student at the Lake Centre as well. And she's actually going further than Ontario to look at permafrost, if you can imagine. She's going to Old Crow Flats in the Yukon. And she's looking at, this methyl mercury that Adam was talking about, which is a more toxic version and it's more mobile. She's trying to understand if the changing landscapes up in Old Crow Flats in their permafrost, if that's actually a problem up there. So very similar, but Nicole partnered up with some students in our program in science communication, and she made this fantastic little 1-minute video about her work and about the relationship that she's building with the communities in Old Crow Flats. So let's have a look at her quick 1-minute video.
I'm Nicole, and my partner is Veronica. Today we'll be talking about the effect of mercury on northern Canadian ecosystems. What is mercury? Mercury is an element that has the possibility of causing serious health effects to humans and wildlife. How does mercury end up in northern ecosystems? Mercury is naturally released into the air through volcanoes, forest fires, and human activities such as coal burning. Once in the air, mercury can travel long distances and eventually fall to the ground through rain or snow. After it reaches the ground, mercury can bind to the plants, and within the north, plants do not fully break down, but instead freeze and become buried in the frozen ground. How does it turn into methylmercury? As the warming climate causes the ground to thaw, the soil releases mercury, carbon, and small living things known as microbes. These microbes combine mercury with carbon to form toxic methylmercury. How does methylmercury affect humans and wildlife? Methylmercury is taken up by tiny animals known as plankton. Fish eat large amounts of plankton over time, increasing methylmercury. Then larger animals, including humans and bears, eat the fish, causing harmful methylmercury to increase in their bodies. What I am studying how the thawing of northern frozen soils in Old Crow Flats increases the potential for methylmercury in the environment. This is an important topic to study because of the methylmercury's potential for dangers to wildlife and humans. Thank you, and miigwech.
That was great. Thank you so much, Nicole.
Yeah, really good video to summarize sort of her work and the work that she's doing in the communities up there. I wanted to just point out the artwork that she used for that, and that's traditional artwork that she's partnered with an artist in that community of Old Crow Flats to showcase their work. So I just thought that I wanted to point that out because it's so lovely. But yeah, now speaking of traditional knowledge and working with communities, Amy, I think you're going to introduce our next video.
This is a really important project that has been going on at the Living with Lakes Center in conjunction with the Living with Lakes Center. It's a larger project called Up North on Climate. And this research, it's many different facets of research, but this research really combines the traditional knowledge and memories of elders in northern communities here in Ontario, and combining that traditional knowledge and memories with scientific information all about weather and climate. And the goal of it is to really help communities build a picture of the weather and climate changes that have already kind of happened and that are currently happening and that we see now every day. And so this combination of traditional knowledge and science really provides a complete and incredibly useful understanding of past and likely future changes that are going to happen in these environments, and with a really primary goal of enabling First Nations in those areas to be prepared and to prepare their community as much as possible now that we know climate change is happening and a lot of these changes are inevitable. So this one of the main researchers, of course, is a good friend of ours, Dr. David Pearson, but you'll also hear from many different researchers and partners in this video as well.
The problem that brings our team together is that First Nations in the North are being more affected by climate change than people in the South. It's co-led with 5 tribal councils and Grand Council Treaty 3, who each have a member on our project's coordinating panel. And the councils we're working with represent 59 communities in much more than half of Ontario, from James Bay to Hudson Bay to the Manitoba border. So a very, very important audience to be collaborating and communicating with. These are projected temperatures for the middle of the century and toward the end of the century. I can point out the very severe increase in the winter temperature, and you'll see along the Hudson Bay coast, temperature increases in the neighborhood of 7 and 8 degrees. These are way beyond any discussion of the 1.5 and the 2 degrees increases for the global average temperature. If you look at the summer map, above 30, into 32 and 33, and that begins to pose heat stress issues, health issues for elders and small children. It also increases evaporation from the landscape, favoring wildfires, increasing severity and frequency of summer storms. Flooding, for example, is becoming more of a serious problem in the north because of late winter rain falling on frozen ground. This kind of weather incident is certainly becoming more common. But their way of life is being seriously affected.
So through our co-led project, we're building capacity in First Nations by co-developing adaptation resources that bring together Indigenous knowledge and science knowledge of climate and the land. These reports put together all this amazing traditional and ecological knowledge that had been collected in our partner communities and combined it with climate change science to tell what we call the climate story of that community. So the idea behind the reports and the matrix specifically was to try to connect traditional knowledge and community observations to climate change risks and impacts, and then finally to adaptation options. And over time, that idea sort of evolved, and we came up with what we call our adaptation framework. So the goal of the adaptation framework is to chart a path to community-based adaptation from step 1, which is learning about climate change science and climate adaptations, all the way to getting adaptations happening in communities.
So based on, those community reports that Kim was just talking about, and everything that, Indigenous knowledge and science knowledge provides, has led us to forming these 8 core themes. And these themes really serve as this sort of overarching structure that guides and houses all of the different topics we cover and materials that we're co-developing. The quick guide is really meant to be a conversation starter and to get the ball rolling and ideas flowing about adaptation and climate change in the community. we feel that the strength of these quick guides really lie in how visual they are. So, you know, when the climate change specialist is talking with someone, that person can really start to see what those adaptation options look like in a community that's familiar or similar to their own, and that they're developed from both observed impacts seen in First Nation communities across the North, as well as those possible future impacts that are based on climate science. From highly visual adaptation infographics and a friendly adaptation framework, to representing science visually and having a knowledge exchange network, together we're building climate change adaptation capacity.
So one of the ways that we hope to help continue to build resilient communities is by co-developing an online GeoHub as a one-stop shop for all things climate change and adaptation. So it would combine the Indigenous and science knowledge, and most of the information will be location or geo-reference. So communities can use this tool to plan their adaptation projects based on local climate observations and projected impacts. So first and foremost, with the permission of elders from our participating communities, we'd like to share their knowledge of the land, both digitally and as videos. And then the other thing that we're doing is an app to allow people to share their stories from the land, be it an impact that they've come across, or maybe you know, some of the ways they're adapting to climate change already, and would hopefully inspire people to think about how they might adapt as well. With this highly collaborative work, our project team envisions First Nation communities continuing to share knowledge while using the co-developed resources to move towards climate resiliency for the benefit of future generations. With that, I give the mic to Geneva. She's the climate change specialist at Grand Council Treaty 3, and she has a special interest in youth climate outreach with her member communities.
We've been involved in a project that really helped set the stepping stones for getting our nation of Treaty 3 moving towards adapting to climate change and mitigating emissions in our region. Our nation is comprised of 28 First Nations all over northwestern Ontario. Not every region has the same impacts of climate change, so we really need to be creative in planning for our First Nations, make sure every community is included. Up North on Climate Group really helped with the development of our projects going forward. We take that knowledge and then we house it within our organization, and we pass it on to our youth, because that's part of our strategy at Grand Council Treaty 3 for building technical capacity within our First Nation and leading our nation to a state of resiliency is investing in our youth and passing that knowledge forward, because by motivating them to make change, we can be a stronger nation when addressing climate change.
Wow, that is so powerful, right? It's just empowering the youth to really, you could hear it in her voice, couldn't you? That she's just really, really grateful for this, for being empowered really to work with the team out of the Lake Center to make some real changes and being able to adapt in their community. So powerful.
And this is a problem, right? That isn't going to happen, 4 or 5 generations down the line. This is imminent. This is imminent for these communities, and an extremely, extremely important work that's going on. Absolutely.
Well, that was we just heard from Dr. Dave and his group, and another veteran of the Valley Living with Lake Center and Sudbury regreening efforts is Dr. Peter Beckett, and he's going to wrap up our show today. So we are running a little bit. Well, we're at 8:03. So those of you who are hanging out with us, thank you very much for sticking around. Dr. Peter Beckett, many of you in Sudbury would have seen Dr. Beckett, just like you would have seen Dr. Dave on either reports about regreening or anything environmental in the city, right? So Dr. Beckett is one of the most important scientists that was and still is absolutely instrumental in the regreening of our city. He's been invited, this is amazing, he's been invited to participate in the United Nations subcommittee, United Nations, yes, the United Nations subcommittee on the decade of ecosystem restoration. And of course, this is thanks to his work in Sudbury and the amazing recovery of our ecosystems, like you talked about already, Amy, the Sudbury method, right? So we're now going to let you listen to about 5 minutes of Dr. Beckett himself telling you a little bit more about this important work and the decade of ecosystem restoration.
United Nations is using Sudbury, basically saying, look what they've done in Sudbury, we can do it elsewhere. So I will thank the fact that Sudbury has changed so much that I'm able to sit on this subcommittee and actually bring ideas from Sudbury directly to the United Nations decade on ecosystem restoration. The major aim of the UN decade is in preventing, halting, and reversing the degrading of ecosystems worldwide. And I don't think it takes us much where we are now to realize that there's a really urgent need to restore damaged ecosystems. Ecosystems support all life on Earth, and the health, and we know actually from work that has been done in Sudbury that the healthier the ecosystems, the healthier the planet, the healthier the people. So they're all linked together. And it's tied not just to the natural environments, but tied to people. The idea that by restoring ecosystems, then we can help to end poverty, we can help to end discrimination, it can help to combat climate change, and actually help to maintain biodiversity or even improve diversity and avoid the supposed next mass extinction. And we all have to get involved in the next decade if this is going to succeed. We need restoration leaders of all kinds, not just scientists, but people who actually carry out the restoration, to get people to carry out restoration. And we've had great leaders at the Limb of Lakes, and we're going to have a great new crop of leaders as the new generation come along. We need to build capacity, and this particularly means working with Indigenous people, both here in Canada and around the world, with youth. And we certainly know here how to celebrate a culture of restoration and to promote restoration. And as always, we need more good science, more information, and so this will be a continuation of one of the mandates of the Limb of Lakes Center and partners. And if we think of fresh waters and ways in which the Limb of Lakes and the co-op can contribute, and has been contributing and will continue to contribute, we can think of fisheries research. We've got our water quality monitoring and assessment. We've got freshwater food contaminants, water land linkages, and lake trout restoration, all of which will be able to feed in to the program. In the area of peatlands, we've got people working on climate change, the permafrost and methane. In urban areas, there's the green technologies, biofuels, pharmaceuticals, waste management, and of industrial areas, and by here I mean not only just tailings, but other kinds of waste management. Communicating what restoration is all about, how to do it. This is a major opportunity, I think, for science communication to be able to inform people about what needs to be done and how it needs to be done. And of course, all the links with the First Nation communities in Northern Ontario and elsewhere, which the co-op has, and hopefully will be able to maintain and sustain those. And then our local restoration example beyond what goes on with the city, and that's the Junction Creek restoration. So I think the co-op going on from here is going to be a major help and charge in achieving the aims of the UN Decade on Ecological Restoration. All we got to do is get on with it and end up with a far better world in 10 years than we have now.
What more can we ask for?
What can we say, right?
What can you say, right? I mean, how inspirational is that? I love Peter's message, Dr. Peter Beckett's message of get on with it. He's basically giving us our marching orders, get on with it so that we can have a better world in 10 years than we have right now.
Absolutely. So that about wraps up our night, and I am so happy that at least I hope that we were able to bring you just a little sliver, a little introduction to some of the just snippets of the amazing work that happens right here in Sudbury, right here in Northern Ontario, and it's a long-term research tradition that not only happens today. I saw in the chat, somebody was saying there's a great crop of leaders that are coming out of the Living with Lake Center. The future is there, and they are building on an amazing tradition that we have had here in Sudbury.
Absolutely. And I would like to thank you, Amy, for being such a great co-host, for being such a great hostess and co-host with me. And I really want to thank Science North for giving us the opportunity to really share our research stories with you and with the entire community, because that's what Science North does. Science North is there to help the scientists and the researchers in our community really get heard, and bring the science down to, to all the families and visitors that come to the Science Center. So we really appreciate that. So thank you very much, Amy and Science North.
Oh, well, thank you. Merci, miigwech. So thank you again from all of us here, both at Science North and the Living with Lake Center. We're so happy to finish off Earth Week celebrating. And what we really want to do is encourage you to go ahead this weekend and do something to protect. Get on with it, as Peter says, right? Do something to protect our land. Learn more about the environment around you. And just remember, here in Sudbury, it wasn't just scientists. Scientists are important. But it wasn't just scientists who made our environmental remediation happen. It was all of Sudbury, the citizens of Sudbury that helped with that as well. And we know we can all make a difference. So thank you, everyone, and have a great night.
Good night.